Welcome everybody to this first roundtable on translational research. It's my pleasure to introduce David Sander, the chair of this session. David is professor at the University of Geneva and the international expert in the field of emotion and cognition. And David, the stage is yours.
Thank you very much, Dominique. Thank you very much, Carmen. So nice that together with Aurélie, you organize this roundtables and this meeting. So we will start with this first roundtable, where we will ask a very important question of translation research. So what can we bridge from research on animal in stress research to research and applications in human research? And so we will take the chance that we have researchers from many disciplines in our association, in our network, so that we can build this bridge together between animal research and human research. And we will have 4 presentations. Each presentation will have a duration of 7 minutes, and then we will have around 3 minutes for questions. And at the very end of the roundtable, we will still have probably around 10 minutes for general discussion around this topic. So I suggest that we directly start with our first presentation. So I will invite Johannes Bohacek. I will not give him the floor, but give him the screen in a way for the Zoom presentation. So Johannes is leading a lab that studies how stress affects behavior through molecular changes in the brain. And his presentation will discuss the molecular signature of a healthy stress response. And Johannes is at the ETHZ Zurich. Johannes, please.
Hello, everyone, and thank you very much for having me. Since I'm the first speaker, I'm going to spend one slide on some general information about stress. So when we face an acute stressor, that is actually a good thing, right? As most of you know, the acute stress response empowers us with an extra boost of energy and attention focus to give us peak performance in situations when it really matters. However, if a stressor is too intense, too severe, or if it keeps occurring over and over and becomes chronic, that's when we're facing an increased disease risk for neuropsychiatric disorders. The point that I am arguing and that my lab focuses on is to say that we need to understand this acute aspect of the stress response in order to make sense of all the other effects that stress has. And there's surprisingly little that we actually know about some of the molecular and circuit details about acute stress. That being said, our lab focuses particularly on the tiny locus coeruleus, the place in the brain where most of the noradrenaline is synthesized. And when you're experiencing an acute stressor, noradrenaline is released into the entire brain. The second structure we're very interested in is the hippocampus, every stress researcher's favorite structure. It's very well characterized. It is very sensitive to the effects of stress, and it is a link to the emotional and cognitive changes that can occur in response to stress. So how do we tackle the question of translation? The one way to do this is what I call translation through technique. The obvious advantage in basic research is that we can manipulate our model systems in a way that we cannot manipulate the brain of patients. In our case, we work with mice, and a classic example is optogenetics. So we can switch specific cell populations on and off at will. And what we've decided to do is to combine this kind of control with readouts that are also clinically relevant. So readouts that you can also employ in the clinic. One example is to manipulate the locus coeruleus with optogenetics and then look at the pupil response, pupillometry, which is something that you can do in patients. That's frequently done in clinical or in research settings with humans. And we can show, for example, that driving the locus coeruleus with increasing intensity leads to an increased response in pupil dilation. Another example that is for translation through technique is resting state fMRI. So this is a brain imaging technique that allows you to assess the functional connectivity across all different brain regions in real time. And again, here we've combined our circuit manipulation, in this case, chemogenetics with DREADS, in this published work, but in ongoing work also with optogenetics. We combine this with functional fMRI readouts, And what we try to get is a fingerprint of how does an altered noradrenergic response look like in terms of whole brain connectivity, and can we maybe recognize this in patient populations or stratify patient populations for those who maybe have alterations in their noradrenergic system. I'm thinking about anxiety disorders or depression, where noradrenaline plays an important role. However, there's also the more basic science approach, that is to do science for the sake of finding out things that we don't know yet. And for this, I'm coming back to this idea of the acute stress response being something good. And what I want to draw your attention to is the healthy stress response. So in most cases, we're facing stressors every day that we can deal with perfectly fine. And we have a model for this in the lab where we throw a mouse into a cold bucket of water for 6 minutes. That's very stressful for the animal, but the day after, the animal is completely fine. In fact, if we screen behaviorally, we cannot detect any differences. So the animals can overcome this stressor for all intents and purposes. So what we ask is, what does the molecular healthy stress response actually look like? And that's a pretty tricky question, because stress happens acutely, and then on a molecular level, waves of events unfold on all different molecular levels. And so we're chasing these changes over time. So I'm drawing your attention to the time course here. And I'm not going to be talking today about all the other things that one can study, like the subregions and how different cell types change. With single cell technology, we have great control over all this. And of course, how the neuromodulators like noradrenaline again impinge on this. For today, and this is my last slide, I'm just showing an example of what the time course of a healthy stress response looks like in the mouse hippocampus. So here we've used phosphoproteomics. That's a technique that for the first time allows us to sample thousands of phosphorylation, protein phosphorylation sites at the same time in the mouse brain. And we see that immediately after stress, protein phosphorylation is the quickest molecular change that we can detect. Immediately after this 6-minute swim stress, there's a very strong response in protein phosphorylation. Every dot you see in red, and these are hundreds of dots, are individual protein phosphorylation sites that are increased after stress, and the blue dots are decreases. And what you can appreciate is how quickly over the course of not even 30 minutes, this response completely dissolves. It's all gone again. And where does it go? It goes to the next molecular level. It goes to gene transcription. So we can look with transcriptomics at which genes are changing. And you see at the 45-minute time point, we start getting a very robust gene expression response in the brain that again kind of wanes and disappears over the course of a couple of hours. And where do these changes go? They go down to translation or translation inhibition, as we've heard today. And so what I think is what we're trying to do is use this characterization of what a healthy stress response is supposed to look like to understand where could, in response to very severe stress, this response go wrong, and how can we recognize this in animals that are maybe at risk. And I would hope that we could translate this to the clinic one point by maybe looking in post-mortem brain tissue to see, do we see similar signatures of dysregulated signaling cascades in patients? Or maybe we could do an approach like this, not in the brain, but in the blood plasma, and compare mice versus humans side by side. With this, I thank you for the attention, and I thank my team, pre and post social distancing, for all their work.
Thank you so much, Johannes. Very interesting talk and very impressive how much you managed to say in less than 7 minutes. Thank you. So I guess we now have time for questions. We have at least 3 minutes. So we can have questions from the panelists. I don't see any questions from the attendees so far. Maybe I can start with one question, Johannes, if you agree. I think it's a question on the approach that you have. How can you select the good stimulus that would be a healthy stressor, in the sense that it's not so easy to have a stimulus that will not elicit, I guess, an unhealthy response? So yeah, how can you select a good stimulus? Because the coping potential should not be too low for the animal, I guess. So how can you select a good stimulus?
So we currently have two approaches for this, and we haven't really selected stressors that are too severe so they really lead to maladaptive responses. But the two approaches are briefly, one, we've now characterized what the healthy response look like. We know which genes should and should not be turned on when. So we can simply use stronger or longer lasting stressors and screen for points of deviation from this normal response. That's one way. And the other way is to use behavior. So we've invested a lot in behavior screening in the animals so we can recognize when do we start seeing maladaptive behaviors. For example, is an animal still anxious on day 2 after a stress exposure on day 1? In the case I've shown you now, none of the animals are anxious anymore. But if we use more severe stressors, maybe they will be.
Okay, so duration of the stress is a key issue. Okay, thank you. Another question, Manuel, you also have a question, I think.
Hi, Johannes. Very nice. Thanks a lot, David. I was wondering whether the phosphor responses that you see and then the delayed transcriptome responses that you see, whether they are causally linked to one another or whether they run into parallel streams, but then they simply have different temporal dynamics.
I'm wondering if you didn't lose Johannes. Can you see him or hear him?
I think he's not connected.
Okay. There is probably an issue with connection, I guess, no? Yeah. This has to happen with Zoom at some point.
Maybe
okay.
I can add, yeah, maybe I can add something here. I'm not going to answer for Johannes, but yeah, I had a similar question because indeed it's quite intriguing. I mean, he put it, he's not here to defend himself, but he put it very nicely that it starts with phosphorylation, which is it's post-translational modification, right? So it's normally a process which happens after translation, but he put it first, and then he argued that this leads, or this leads to transcription, which then will lead to translation to produce the effect. That's exactly the question that you asked, Manuel, whether there is any causal link and why would phosphorylation be needed for transcription? It would be needed to phosphorylate transcription factors, for instance, or factors, signaling factors. But this, in temporally speaking, I'm not sure if this would fit really in this very narrow few minute window that he's looking at. Another aspect is that protein phosphorylation is it's a constant. There is homeostasis of phosphorylation of some factors, so you don't need necessarily to produce it to get activity of the proteins.
Agreed.
I just received an email from Johannes saying that his Zoom just crashed, so he will be back soon. Maybe we can ask him again if we have time at the end of the roundtable what he could add, if it's okay for you. So I suggest that we go directly to the second presentation of this roundtable. So Christophe Lamy, I invite you to share your screen. So Christophe is now at the University of Geneva, and his lab is interested in understanding the very basic neurobiological mechanism underlying emotion, cognition, and behavior. And he's particularly interested, as we will see in his presentation, about interoception. Christophe, please, the floor is yours. We don't hear you, Christophe. Your microphone is on mute. Christophe, we don't hear you.
Better now?
Perfect.
Okay, sorry for that. So thanks for the introduction. So in this presentation, due to the time limit, I will just briefly outline my research interest on the role of interoception and its effect on brain circuits, and then try to elaborate a bit on our strategies that we are developing now to translate this to the patient.
And share our screen. So please, Isabelle. So Isabelle Mansuy, she's both at the University of Zurich, and you are also at the ETHZ Zurich, and your lab is interested in the epigenetic basis of complex brain function. And we are very much looking forward to your presentation, Isabelle, please.
Yeah, thanks very much. for asking me or offering me to participate to this roundtable. My presentation will be a little bit different from the previous one. It's not going to be a research talk. I'm not going to present any data from the lab, but I thought I'll just share some ideas, some concepts to feed the discussion or the thinking about translational neuroscience, how we can really move from animal models, data, quantitative data in animal models, and possible therapies or better understanding of the consequences of stress. And it's fairly new in the in neuroscience in general to consider epigenetic factors as important factors at the interface between the environment and the genome, because it's really a interaction between the genetic code and experiences in life, environmental factors, which really give rise and cause symptoms and phenotypes and control our mental and physical health. So I'm not sure how to get rid of this window here. Okay. I guess you can you see the window is also on your way, or?
No, we don't see any.
You don't see it. Okay, good. So epigenetics is the ensemble of factors and mechanisms that regulate the activity of the genome. And beauty, unlike the genome, the epigenome is modifiable by environmental factors, by life experiences. And when we are exposed to stress, whether prenatally, during early life as a child, or later in life as adult, or even in old age, this can activate or inhibit epigenetic processes and change epigenetic profile across the body in any cell basically of the body, including germ cells. And that's what I'm interested in. My lab is working on epigenetic factors in germ cells for the transmission of the effect of stress. But so what are the epigenetic factors that we can consider? I mean, there are many epigenetic factors, but there are a few which are easier to approach because we can measure them, we can identify them, we can measure them. And the three main factors which today have been studied in the context of stress, mostly in animal models, but also in patients, are DNA methylation. So these biochemical modifications is the addition of a methyl group on the cytosine of DNA, which decorates the whole DNA in both encoding and non-coding regions. Histone modifications, so post-translational modifications on protein histones. I mean, there is a histone code. There are many different types of modifications which combine each other in a very different way, and which are combined with DNA methylation, and the methylome plus the histone code are together interact to regulate the activity of the genome. And another type of factor which may fall in the category of epigenetic or not, it depends on how we use the semantic, but factors which regulate the genome and which have also been shown to be altered, modified by stress, are non-coding RNA. Non-coding RNA which exist in the cytoplasm, but also in the nucleus, and they are small non-coding RNA, microRNA, pRNAs, no-RNAs, and long non-coding RNAs. So there is a whole panel of epigenetic factors which are present in neuronal cells, in glial cells, but also in other cells, which may be linked to the comorbidities with stress, because stress does not only affect brain function, so behavior. Stress also affects physical health, and there is a link between stress and cardiovascular diseases. Mohamed showed us the link between stress and cancer. And there is also a link between stress and bone health. So there are many links. And which could, in the body, which could be explained by the fact that epigenetic factors are everywhere. So where and which epigenetic factors can be measured in humans, and where and which epigenetic factors could be considered as potential markers for stress? Blood is the most accessible biological fluid that can also be obtained from psychiatric patients. CSF is another fluid, but it's much less accessible, and in conditions of when the transmission of symptoms are considered across generations. Sperm is another type of fluid which has also already been started to be analyzed from an epigenetic perspective. But in blood, so you can measure DNA methylation, you can measure non-coding RNAs, microRNAs, but also other non-coding RNAs. In blood, RNAs are not really freely floating. Many are present in EVs, in extracellular vesicles. So there are now many people developing methodologies to isolate EVs and do RNA sequencing from EVs coming from blood or from serum. The problem with this is that blood is a quite heterogeneous liquid. There are many different cells, from red blood cells, but white blood cells, and each of these blood cells may have its own epigenome. So some people have used whole blood, but you can also use PBMC, so white blood cells in particular. However, the signal that you may obtain may not be that clear, and in that respect, more work would be needed to identify the epigenome of each cell type, depending on the status of the individual as well. The other question is, what is the correlation between an epigenetic factor like a microRNA or DNA methylation profile measured in blood or even in a subpopulation of blood cells, and what happens in the brain? So there are studies which have been done in mice to really try to find whether there is a correlation, and it's not an all or none response. If you look at some microRNAs, you may find that they are dysregulated similarly in brain and blood, depending on which brain area you look at, but some other microRNAs or other DNA methylation sites, they will not be dysregulated the same way. There is the option of cell-free circulating nucleic acids, RNA or DNA, which are present in blood, but they are released from different organs, including the brain. So if this, the cell-free circulating RNA or DNA coming from the brain, if they could be identified in the blood, that may be a possibility for more specific epigenetic factors. How about epigenetic or epidrugs? Epidrugs, they are drugs which act on different enzymes of the epigenetic machinery, DNMT, so DNA methyltransferases or histone deacetylases. There are a number of drugs. I put just a table here of drugs which have been used in cancer therapy. Because oncology is quite advanced in terms of identifying the epigenome of cancer cells or tumors and using epigenetic drugs. Some of them are already in clinical use. However, the complexity of this is that unlike in cancer, where you have a tumor where most tumor cells may have an increased DNA methylation, for instance, let's say 80% increase of DNA methylation, with complex pathologies like those induced by stress, it's not like this. There may be increase in DNA methylation at some places and decrease at other places. This has been demonstrated already. And there may be more microRNAs in one brain area and less in another. So there is no single drug which could fix the problem. So that's a difficulty. And that's why considering one drug, I mean, it's a little too premature, and more research would be needed for this. Maybe in combination with psychotherapy, or or developing more specific drugs which don't act on the major enzymes like this, but maybe on bromodomain proteins which are associated with these major enzymes. Maybe very quickly, a last thing, it's just an idea because it's very innovative. So it's the use of CRISPR-Cas, the inactivated version of Cas. So the D is for deactivated or dead, which when it's fused with another protein, let's say TET, DNMT, or whatever, a bromodomain again, can allow to target, to modify specific epigenetic marks on a given locus. So you have guide RNAs, which guide the DCAS, which is in blue here, to a specific region of the genome. And if this DCAS is fused to the enzymatic activity of TET, it may induce demethylation of the locus which is next to it, or if it's used with DNMT, it will increase the methylation of this. So this has been shown to work in vitro, also in vivo, in the brain, in mice, when viral vectors expressing this version of Cas are expressed, are produced, but obviously in human, we are not there yet, but this type of technology in the first step could be used to gain understanding about the causal role of some epigenetic factors or some epigenetic alterations, gene expression and symptomatology in correspondence with what was observed in human in patients. So I think I will stop here. They are just general ideas, but yeah, I hope that
very useful.
Useful. Thank you.
Very useful. Thank you very much, Isabelle. Thank you. Maybe we still have time for one or two questions that we can take for Isabelle. These are quick questions. Maybe I start with a very quick one. Isabelle, is it about acute stress or about chronic stress? Mainly that you suggest that these epigenetic factors could be measured also in human.
I think it's both. It's both, because it's known that a stress which has happened in childhood can leave traces on the epigenome that can be measured in adulthood. Some epigenetic factors can be altered permanently, and even in the following generation, actually.
Okay, thank you. Is there one question maybe from the panelists that you would like to ask? You can also save questions for the discussion at the end, but otherwise, please feel free.
No, I think Isabelle gave a very good overview of the potential also of all this basic science. So, because the question of that you're addressing here is indeed if your science is contributing. So in this case, I think you are very positive, so much, not so much for the drugs, because as you said, it's very complex and targeting has to be more directive somehow. So my question is a little bit like sometimes the journalists have these questions, no? How many years you think the people or patients need to be waiting for this to be effective now?
I can't really tell, and indeed, I think I would rather be putting forward words of caution. I would myself not really consider epidrugs as ever being a possibility for complex disorders.
No, for the CRISPR, the DCRISPR that you
for
propose.
the CRISPR, it's the same. I don't know. It's difficult to predict because, you know, sometimes you think it will take time and things sometimes you can develop very quickly. There is not only the science, but also there is all the ethical matter. The beauty of the CRISPR-Cas system is that it's not changing the genome, unlike the classical CRISPR-Cas. So there it's also reversible. So that's probably one of the power of this system that may make it applicable to the human beings one day in the future.
Thank you.
Thank you. So the question of what are the traces that stress will make is also a question that is of high interest for Manuel Mameli, who is our last speaker for this roundtable. So Manuel is at the University of Lausanne, now at the Département des Neurosciences Fondamentales, and his lab is mainly interested in how the brain reacts to aversion and to reward. And Manuel, we are very happy to have you as a speaker on the roundtable, please.
Thank you, David. I also join my colleagues in thanking Carmen and Dominique for putting together this roundtable of discussion around stress. So I'd like to complement the vision that Isabelle and Johannes and Christophe provided previously, adding up another neurobiological process that is vulnerable to stress conditions. And this is namely synaptic function. I mean, we all know that in neurons in the brain, presynaptic terminals that are invaded by an action potential lead to release of neurotransmitter, in this case depicted here as glutamate, that then interacts with a specific protein located in this postsynaptic compartment, which in this case are AMPA receptors or NMDA receptors. In other words, we know that the synapse is the unique feature that provides a neuronal communication, like is the fundamental process allowing neurons to talk one another. Now, the weight of this communication can vary. So the strength of the synapse is extremely dynamic and extremely plastic. So it can vary over time, but it also importantly varies upon experience. That can be a developmental experience, or it can be an input from the external world. And this process of dynamic change in neuronal communication at the level of the synapse is named indeed synaptic plasticity, which has been thought of to be one of the cellular correlates, for example, for learning processes. Yet, synaptic plasticity also can be aberrant and can be not properly occurring. And this is indeed the case, at least in the context of today, when stresses arise. And as Johannes mentioned previously, especially in this case, a persistent chronic stress, including long traumas or bullying over time or early life stress, can not only release stress hormones, but then a cascade of events lead to aberrant form of synaptic plasticity that takes place at this synaptic level. Now, in the last 5 years, the development of technologies allow us to tackle specific synapses. And why this is important? Because together with the aberrant synaptic plasticity, we also have the emergence of stress-induced pathological states, namely depressive states, cognitive deficits, or aspects of post-traumatic stress disorders. So what am I talking about in terms of neurotechnologies? In a span of 5 to 10 years, we discovered that optogenetics, for example, can allow us to target the presynaptic compartment or the postsynaptic compartment and change the extent of synaptic transmission. The same is valid for the brain stimulation that allows us to change the strength of a larger network of synapses. Similarly, this is valid for pharmacogenetics that can allow us to tune up or tune down the effectiveness of this synaptic connectivity. We don't have to forget about pharmacology. So running through mechanisms, we can target a specific protein and increase or decrease the strength of these synapses. And last but not least, just published, this approach called living electrodes, where we can create experimentally a network of neurons that we can manipulate, and their manipulation allow us to control and tune synaptic transmission. Why is this important? Because in the last few years, we found that by normalizing with such a range of tools, synaptic efficacy after stress, we also normalize certain behavioral aspects, the depressive states, the cognitive deficits, or aspects of post-traumatic stress disorder. Therefore, arguing for the existence of a causality between the synaptic plasticity changes after stress and the pathological changes. Now, the picture like this will be beautiful, but then we incur in a problematic that these are local interventions. These synaptic interventions are very limited to single structure. And we know that the stress alters synaptic transmission in the cortex, in the striatum, in the midbrain, in the lateral avenula. So how do we come up with a strategy such that we can use this information at the synaptic level, but then now enlarge a little bit to what happened to the brain and to the timing by which stress produces a modification. And what I think is happening is that we are in a sort of a transitional research momentum where we need to capitalize on what we know about synapses and incorporate it in a wider way. One way I'm referring to is to use maybe technologies that are like the following: brain-wide fiber photometry or neuropixels, with which we can study at the same time the activity of multiple structures. Then match it together with behavioral machine learning, for example, so allowing it to study specific behavioral readout very precise in time. So because like this, we can reach information like which is the locus at a given time that is time locked with a specific behavioral alteration that emerges after stress exposure. And I think, again, capitalizing this synaptic investigation and the possibility of several interventions, together with the brain-wide investigation in rodents, we can also match those human investigations that tell us about structures that are implicated in humans at certain timing. But in order to do so, I think we need to create overall clusters of interaction between clinicians and fundamental scientists in order to eventually generate in the future viable intervention with a more cross-talk communication. And with this, I'm done, and I'm happy to take any question. Thank you.
Thank you very much, Manuel. Maybe I guess a quick first question that is a complex one, of course, but what is the major challenge that you think there is to bring the interventions that you have in rodents to human? What is the big challenge? Is it conceptual? Is it only methodological? Is it a question of measures? What is the highest challenge?
I think the challenges are many. I think I will start. Ethical and security is one main challenge, right? We need to come up with tools that allows to be the least invasive possible, but at the same time ethically correct. Now, I mentioned the deep brain stimulation and the pharmacogenetic for a reason, because one is already employed, yet invasiveness. But the pharmacogenetic, there are studies and a strong effort to render this gene therapy available. Now, these are two parallel roads where maybe we can get some results. So I think the design and the ethical is still a barrier.
Yes. Okay, thank you, Manuel. Maybe one more question to Manuel before we open a brief but still general discussion. Okay. Is there maybe a question from someone in the panel to Manuel?
There is a question from the participants, Raisa de Castro.
Yes, okay. Thank you. Sorry, I missed it. Thank you for the presentation. In the context of aberrant synaptic plasticity, what is your opinion on long-term changes in glutamate clearance, which involves much more than just neurons, but also glia cells? Do you think we are only looking at half of the picture, or it has been enough to correlate with behavior changes? From Raisa.
So, I mean, this is a bit of a provocative question. Of course, I don't think we are looking at only half of the picture. I think we are looking at one side and a perspective, which does not rule out the other player. And so it's as if you were to focus on glia, and then you say it's half of the picture because you don't consider neuron. I think it's the composite response is very big. So altering neuronal communication engages alteration in glial function and vice versa. So I think what you are pointing out is very important in terms of now that we know a lot about synaptic communication, get to know how the glial portion plays a role in this function is a next step that is already happening. I mean, there is not anymore the time of neglection of glia in the field. So I think we are slowly but getting in there into having an integrative approach. But I don't think it's half of the picture only.
Yes. Thank you very much, Manuel. Thank you to all speakers. I think despite the fact that we did very well on the timing of the presentations, we don't have that much time for questions and discussion, but still, maybe we can take one or two questions. Maybe I will start with Johannes because your Zoom crashed and we didn't really have the chance to discuss with you. But the question that we have from someone from the public to Johannes, I think we can all think about it, and maybe this can be the start of the discussion. It's a question in terms of prediction. Do you think you can predict from the beginning of a stress response, and in rodent, but maybe also why not in human, can you predict whether the response will be healthy or whether it will be maladaptive? And I think this question of prediction is also interesting from rodent to humans in a way. Can we predict what kind of response that we have in rodent could be the kind of response that we have in human? So maybe Johannes, if you would like to start with this question of prediction.
Yeah, it's good to be back. You should have seen my stress response. Also a good way to evade questions. That's a tough one. I think we all believe that we can predict, right, which individual will have a healthy or a maladaptive stress response. But I don't think any one of us is there yet, right? But it's a somewhat deterministic view that we think somehow our genes and epigenomes and our behavioral patterns predict how we will behave in the future. I think that's the way it works, but I don't know. What do the others think?
Good question. What do you think about this issue of prediction? Okay. Maybe I don't know if someone has a strong opinion. Otherwise, I'll just have maybe one question that I would like to ask to the floor.
Can I say something? I think it's really difficult to predict, right? Because the response may be cumulative. The way you approach a stress or a challenging situation at one point may be very different to the way you may approach it a couple of years later after having experienced different things. So I think it's a dynamic response, and even in psychiatric patients, even in people who have been severely traumatized in their childhood, it's very context dependent.
Mm-hmm. Okay, thank you. Maybe we will, before closing the roundtable, maybe one question. If you had to think of one marker of stress, if you think it can exist, a marker of stress, what would be this marker that you could find across species, both in rodents and in human? What would be this marker if you could think about one that we should focus on for this translational research?
I was hoping for my internet connection to break up again, but my best guess would be, and I'm pointing towards Carmen a little bit for this one, is somehow brain energy metabolism. I somehow think that it's all about energy, and the stress response is about energy. So somewhere in there, not a specific biomarker, but something in how energy is metabolized or produced. That could be an interesting topic.
And if I may add on it, because I agree with Johannes a little bit, it's one of these global things that for sure happen with stress. If only we could start generating tools that would allow us to dynamically control for these type of responses, as much as we have them for, say, neuronal activity, to mention one, that would be a great advantage. I don't know whether the others agree or not.
I would like to argue that epigenetic signatures would be a great possibility, because that would not be only one factor, but a combination of factors which would be specific to a certain response. Metabolic markers, yes, I agree with you. However, there are many metabolic diseases. So whether you would be able to distinguish this from a metabolic condition, which are comorbid to a psychiatric condition, that may be a bit difficult with markers like microRNAs or different type of non-coding RNAs. Perhaps that could be made brain specific, or at least be measured in blood, reflecting what's going on in the brain, but be a specific response.
Perhaps if I can add something, I think indeed it goes, the field is evolving to stop considering only one marker, perhaps.
Mm-hmm.
Yes, for me, if I made probably
similar to the polygenic risk score. I'm gone now. I'm back.
Yes, you're back. You freeze for a while.
Yeah, I think perhaps a combination of markers and with the use of machine learning techniques, because we are very diverse, and then it should be several markers that eventually are good at predicting in the general population. Probably one marker is not going to be the best one, but I think we are going to see a lot on that in the next few years, probably.
And maybe
yes,
Christophe,
yes, in fact, the stress response, I mean, we consider stress as one thing, but it's a set of responses to different stimuli with different type of subcircuits. I doubt there is one marker. Probably the marker we should look for are the markers of susceptibility, as referenced with the first question, basically, or how an individual would respond to one stressor, and can this predict the pathology that could be generated by this exposure to a stressor. Obviously, not everyone will respond the same way to the same stressor, and I think that's the key factor. I mean, if you talk about translation to patients.
Thank you very much, and we see that there will be many occasions in the next years, Carmen, for the next meetings that we can address these issues of translational research. So I think we need to end this roundtable because the next roundtable will start in a few minutes. So thank you very much again to all our speakers. Thank you again.
Thank you, everyone.
Thank you so much. It was a great roundtable. Thank you also, David.